So I'll put this in the context, of course, of Dmitri Belyaev, because the meeting is called this way, and because I've been visiting the fox farm yesterday, and that was very interesting. That's also why a couple of people are not here yet, because they've been captured. They opened the wrong cage in the fox farm, and they got the aggressive one rather than the pleasant one. I'm talking to you through Manchester and Amsterdam. That's where I'm located. And lots of friends I have over there, and they've helped with this presentation, just for you to realize. I'll be talking about what systems biology is. Then I'll talk about some of the principles. I think that's important for science, to discover principles. That's what we're trying to do with systems biology. I'll give a classical example. I'll give now an example I think that's new and old at the same time. Belyaev did that long before we understood what systems biology was. So that's a bit funny and interesting maybe. And then I'll tell you about something about recent progress, as you can see. That's all right? Any votes against this? Want to do something else? No, that's it? Okay. Thank you very much for the support. So what is systems biology? And what is bioinformatics? Because it's very similar, we think. Now, I think it's fairly easy, and I talk a lot with bioinformaticists in the infrastructure for bioinformatics, and they would agree to this, that bioinformatics goes from data to information, and then systems biology goes from information to understanding. And that, in many areas, would seem to be a small step from information to understanding. But as we've seen in the presentation by Dr. Polly earlier on, in biology we have this non-linearity. The maps between things are not linear. So that essentially means it's not easy to look at the genome and then immediately interpolate or extrapolate and to understand biological function, because it's non-linear. That means a little change in the genome can have an enormous effect on function, or an enormous change in the genome can have a small effect on function, and you don't know beforehand. And that makes this step difficult, as I say, and therefore relevant. This systems biology step is relevant because of this nonlinearity. It's also another reason why it's relevant, same sort of thing. If you think about it, the DNA in all your cells is the same, but all the cells are different. From some perspectives, they're very similar because the DNA is the same, but functionally, of course, in terms of biology, they're extremely different. Don't put a nerve cell in place of a heart cell, because when you're thinking too much of this nice young little girl that's moving around, then your heart starts beating really too fast, right? So that kind of thing, at least for the gentleman, that would be a difficult thing to do. And then, of course, there's the famous Russian history with Belyaev, Zabotinsky, Ilya Prigozhin, and Boris Holodenko. Who's now in Ireland, a very good friend of mine, who've been working on self-organization and bistability and stability, and that is also important here. Basically, if you're doing developmental biology, you'll find there is self-organization, and this network just goes sort of by miracle into a certain state that you cannot linearly predict from just the DNA sequence. So that's where systems biology comes in. So bioinformatics is important for the first step, systems biology is important for the second step. Now, why systems biology? Well, I think I have this thesis that all biological functions are network functions. There's no biological function that is not a function of more than one component. Maybe there is a small mistake. Maybe some of you can think of one biological function that is a single molecule function, but they're going to be few. So this is like a law of nature. Most functions are network functions. And this, here I took a network. Actually E. coli, but it stands for a general network. This is a function, an impaired function somewhere. This function needs a commodity, say energy, and the energy it needs can come from three routes. It could be ATP delivered by glycolysis, ATP delivered by the TCA cycle, ATP delivered by the pentose phosphate pathway, if you want to be technical about it. Yeah? So it's something you need for this function. So if that function is impaired, this could be due to the fact you would think that this pathway cannot make ATP anymore. The mitochondria cannot make ATP anymore, cannot deliver this function. But because there's three pathways, there's redundancy, you get multiple functions, you get multiple causality, threefold causality in this example. But it's even more complex than this, because this cause, if you have a problem here, to make ATP here, this could be replaced by a problem here or a problem here. So this cause could be any of a number of causes along this route. So you have an and an or causality problem, so it becomes complex. I mean, I wouldn't be able to follow what I'm saying here, but anyway, you could see it's complicated, therefore. It's again not linear. Yeah? And that is making things difficult. You get multiple causality. Sometimes these arrows, they turn back on themselves. You get circular causality, and that explains multiple factorial disease to a significant extent. And in fact, Westerhowiąฒ and myself and a couple of others proposed cancer to be a systems biology disease. This was well before Bob Weinberg made the second map. And I think that's much of the essence. And that's why we're so slow in getting further with this disease. And I would say that virtually all diseases are systems biology diseases. And this may tell you something about what I think about the missing heritability problem we heard about this morning in two talks. One by Baranov and one by Stepanov. So, what is systems biology? And we decided to be democratic, of course. And we got a couple of people together. This was done by Lilia Alberghina, who's in Milano, and myself. We got a group of people together to tell us what systems biology is. And this is in this book. It's already a while ago. And what came out is this. The science that aims to understand, it's sort of saying the same as science, principles governing how the biological functions, that means all biological functions, emerge or arise from the interactions in the biological network. And there's a couple of funny things in statements here. First, you see the definition doesn't have mathematics in it. So mathematics is needed maybe for this, but it's not the aim. The aim is the understanding of how you get function from interactions. The second thing, it's definitely about networking. The third thing, it is the biological functions, because all biological functions of an organism are intertwined. They are sort of related. I can walk because I need to eat, and for eating I need a heart, and I need to eat because my lungs have to move, etc. Yeah, that kind of thing. Everything is connected. So you're not often not solving for one function, understanding one function, but simultaneously understanding more functions. And it's the networking, and that's also relevant because actually a living organism only has one network. It turns out that it's not, although Lazos and Georgi has found that the networks are all connected. It is not so that our genes have sub-networks that are completely unconnected. Everything is connected with everything else. So that's important. That also leads to the complexity, and this leads, of course, in medicine to precision medicine. Has been called precision, personalized, individualized, 4P by Lee Hood, and PP4M this morning. And it also leads, of course, to precision biotechnology. And there's lots of potential there for new things. So that is what it is, I think. You will probably disagree, doesn't matter. We can have a fun discussion. But the point is made. Note, it's not defined as mathematical biology. There's a purpose. We need to understand what networking does for biological function. And because networking is complex, you're often helped with mathematics. But maybe not always. Okay, principles. It goes after principles. So we talk about I'd like to give examples, and one of them is a very classical one, done by Henrik Kutcher in Edinburgh. It's the lack of dominance. This is relating to genetics, because the meeting is a bit about genetics. And of course, the second one was more challenging for me. This was Belyaev's findings. And then I recognized that this actually said something about systems biology. So I'd like to present my thoughts on that, and you can discuss it. But let's start with Henrik Kutcher. He was a geneticist, but also a bit of a mathematician, and he was with Conrad Waddell in Edinburgh. And he discussed the recessivity of most lack-of-function mutations. If you have a mutation that is a knockout, and the function disappears, so the thing doesn't function anymore, I can basically, I have two legs, I have a mutation in one leg, then I'm a heterozygote, I have a mutation in two legs, I'm a homozygote negative, and I can't walk anymore, right? With one leg, I can walk a little bit. That's type of genetics. Just for the biochemist, I'm a biochemist, you need simple explanations. We need simple explanations. So that's what we're talking about, and it turns out that most lack of function mutations, if you have them homozygous, of course they kill the whole process. Function is gone. But when you have them heterozygous, only 50% of the genes, you don't even notice them, or you hardly notice them. And Henrik, he was a brilliant guy. First of all, they made him chair of the parking lot, so he didn't have to do any bureaucracy as a professor. And then he could think. And they thought, this is strange. You would expect that if you have 50% of the genes, you have 50% of the function. So he made that remark. So this was the observation. Just to repeat it, this is the F0 of the one type. This is the F0 of the heterozygous, homozygous knockout. This is 100. This has 100% function. The other parent has 0% function. And the observation is that the F1 gets 95% function. So it still functions. And you would have expected maybe 50% function, because if you think about single molecule, if you think single molecule things, then if it's a single molecule, this gene, then you have AA here, you have 0, 0 here, so that's nothing here, 0 function here. This should have been a 0, I apologize. And then we go to the heterozygous state, and then here we have 50% of the function. That's what you would expect from a molecular explanation, single molecule explanation. That doesn't work, that's a linear explanation. The reality is 95% only, or 90%. Yeah. And Henrik Katje found a systems biology explanation that comes with this, which you will probably agree. If you would think about a function, and this is an enzyme amount, or a gene dosage, then if you have very little of the gene, you get some function, some flux through a pathway. And if you have more of the gene, you get more of the flux. But if you get, once you get more and more of that gene, this gene becomes excess. You have an excess of these genes. The other genes start to limit function. So there's a maximum flux through the pathway that is not determined by this gene number 1, enzyme number 1, but it's determined by the other enzymes. So it looks like this. And you will notice that this is, again, this nonlinearity. This is not a straight line. And you can see it is completely obvious. So this is obvious. This happens all the time in biology. So biology is always nonlinear. Now, you can define a measure of the extent of dominance here. And that is actually how strongly function depends on activity, gene activity. And that is basically the change, the slope of this line, how the flux depends on that gene dosage, is the slope of this line. And for those of you who know a little bit of mathematics, the slope you can do by taking the derivative of J for flux, over enzyme. And this is done relatively. So you divide this by J over enzyme itself. So you get the relative change in flux for a certain relative change in gene dosage. So basically it's the percentage change in flux for a 50% reduction in gene dosage. If that is 50% reduction of flux, you get a C of 1, a dominance of 1, complete dominance. If you reduce the function by 50%, then you see a reduction. So if you reduce the gene dosage by 50% and your function goes down by 10%, and you get 10 by 50 is 0.2 dominance. That's the type of thing. Anyway, this will become a bit more clearer to you, but except that this is a measure of dominance. And we have done this for a model we made a very good model, completely experiment-based, database. You can see these dominance coefficients, or control coefficients, they are indeed not 1. Only one of them is close to 1, but it's still 0.6, and the others are very small. So none of the genes seems to be dominant for this particular function, which is ATP synthesis. You may see something like the sum of all these dominance factors is 1, and that is actually something that leads me to a principle, because by using systems biology, you don't only have these observations, but you also have some mathematical proofs. It's not only applied mathematics, but also fundamental mathematics that you can use. And by using this, you can actually prove mathematically that the sum of these dominance coefficients must always be equal to 1. And you can prove this mathematically solid. And mathematics means that there's no escape. So it means could be convincing. It's often not convincing to people because they want to see an experiment, but it just happens. Mathematics can be, there's no escape. So, what is relevant here, and that was what Henrik Katje did, he realized that the implication of this is that the average, if you sum n things, like 10 things, and they must be equal to 1, then on average there will be 1 divided by n. If you sum 10 things, and their total is 1, then they are on average 1 divided by 10%. And 10% is small, not for physicists, not for mathematicians, but for me as a biochemist, 10% change is small because I can't measure it. I'm not accurate enough. So 10% is smaller, so that's what this tells us. So this is going to be very small. So he realized that this, for a long pathway, say 10 genes, it's always going to be small. Now we go back to this, and hopefully you'll recognize that what you then predict, if you do this, you go to this heterozygous 50% loss of function for a 10 pathway enzyme, the 50% of function only becomes 5% loss of function. You go back to 95%. And the heterozygous is like the wild type. So that is an illustration of how Henrik Kutcher explained an important phenomenon in genetics, got around the non-linearity of biology by this approach. So it's a marvelous systems biology approach. I can also explain it this way. So this is basically now the same, this is a pathway here with different genes. And here I have the knockout. This gene here is absent in this parent. Now I do the heterozygote. So I get this situation. You don't get this situation basically. When you get half this gene dosage, you get only half the flux. Because what happens is that although you have half the gene dosage, these molecules that are being produced here are heaping up, are piling up. You get a high concentration here. They push more flux through this enzyme, and you get a compensation phenomenon in biology typically, and you go back to 95%. So that's something that we understand now, right, by systems biology. Now we go to co-selection by Belyaev, which I found very interesting. So that's, the other one was the Edinburgh example of principles. Now we have the Novosibirsk example of principles of co-selection. And that's the phenomenon that selection for domestication also brings drooping ears, and that is what we have here. So basically we see that we start with this, let me put it this way. We start with aggressivity and upright ears, and both of these are coupled or are encoded by their own gene, and now we have a knockout of this gene, and you will understand that you get domestication, loss of aggressivity, but there's no effect on the ears because the ears are encoded by a different gene. So you would not understand that there is this co-selection of ear form with domestication. But if you look at it like this, and the pathway is like this, and now you have this pathway leading both to aggressivity and to upright ears, then by selection you select for a mutant that has less of this, then you get simultaneously less of aggressivity and less of upright ears. So you will simultaneously get domestication and drooping ears. And here the explanation is that basically what Belyaev was seeing and was understanding was a network effect. He was understanding and seeing network effects in biology. And we saw that in Professor Popopa's lecture that this is actually what was the further thinking also. It's a network like this, which is very similar to what I just showed. You find that if you do a change here, it affects all the downstream pathways in there. So that is the second principle example. Now, I'd also tell you a little bit about the progress, and one of them is the Make Me By model. And basically what we're doing now is we have an infrastructure set up that allows any group in the world to ask us to make their model. So if this is an experimental group, we can make a mathematical model and we can solve these type of issues. So that's interesting. Alexei Kolotkin will talk more about this. Second thing is the genome-wide metabolic maps that I'd like to tell you about. You may think that the genome, with its 25,000 genes, the human genome is extremely complex, and it is. But it doesn't mean that it's hopeless. You can play certain tricks, systems biology tricks. And I'm a bit proud that together with a rather vast network of scientists, we've been able to do this complex human network of genes, looking at the metabolic genes to make a consensus metabolic map. So this is shown here. Actually, it's a simplified picture of the true map. But we here have the map of the total metabolism of the human. This means all the enzymes of the human and all their enzyme activities are written on this map, are connected. So this means you can now see what we can make as a human from food, from, say, hamburgers, what we can make from hamburgers. Anything, any molecule we can make from hamburgers you could see here. But maybe there are places on this map that are not connected to food 1, which is hamburgers. So this would mean you can not only eat hamburgers. This is correct, as we know. So you can see that, indeed, this map can serve to tell you what type of food you have, what type of food you should eat. But it also tells you about individualized medicine. If I have a mutation, a single nucleotide polymorphism, and I'm lacking this gene, or say this gene, then it may be difficult from this food to make these substances. So I will have a problem in my developmental biology. I can't make these molecules anymore. So you can then see that using this map, we can say, well, why don't you take this food, food number 3, is maybe hot dogs. So you take hot dogs and then you can still thrive. Yeah, that's the principle. Does that work? Yes, it does, because we know this already, and we've replayed this. This is for phenylketonuria, a known inborn error of metabolism. We can now completely understand that if, so this is phenylalanine, this is tyrosine. If you have a deletion of this gene, you cannot make any tyrosine anymore. There's not enough tyrosine normally in the food, so therefore you cannot live. And you can also say what type of this pathology, therapy you would have that is enhanced tyrosine in the food and reduced phenylalanine. How much time do I still have? 5 minutes, thanks. 3 minutes, okay. 3.5. I like the half. Okay, so now of course we're wanting to do this kind of thing for the serotonin that we heard so much about. We want to do that mapping, right? And that is now an entire possibility. It's fascinating because it links the biochemistry to the mind, of course. And then there's epigenetics and noise, and there's lots of things I'd like to tell you about. For instance, that we're measuring this, and we have these beautiful models about yeast cells, and we suppose that in these models they're homogeneous. This is the amount of glucose uptake into cells, and you can see here there's lots of cells that have 50% of the cells or 40% of the cells have a lot of uptake of glucose, and about 50% of the cells have much less uptake of glucose. All these cells have the same genome, so this is now cells with the same genome and different function. And that is then phenotypic heterogeneity, and we're discussing this, we're looking at this. There's various explanations for this, but in this case, it's the same genome, so that's not happening. We're doing these in the lab, so that's not happening, external conditions. So we're talking about epigenetic diversity only, and this could be by stability. It's not, we know that, and it's intrinsic noise. So we're now modeling noise in transcription, and that comes with epigenetics, and it comes with a lot of things that I don't have time for at the moment, but it also comes with why it's relevant. And this is important for breast cancer. Breast cancer, where much of this is estrogen receptor positive, so that works like this, that somewhere here there's an activation, so that there's always proliferation in this part of the breast tissue. This then becomes the tumor, right? But this is still activated by testosterone, which is converted to estradiol. And the strategy now is to have tamoxifen that interacts with this activation here. So that's a standard treatment that works very well. So it works for, it induces remission in all these breast tumors. But the problem is that after a while you get half of the patients develop resistance. And then the question is whether all cancer cells become resistant or only some. And what you see is that it's a subpopulation of all these tumor cells that become resistant. So we're looking into this, and you can realize that, for instance, this is one of the things where we can see that this happens. Here we are staining for DNA, so that tells us the 4 nuclei. We're also staining for glycerolide 3-phosphate dehydrogenase, a metabolic enzyme. That's in red. All the enzymes, all the cells have it. And we're also staining for CD44, which is related to this drug resistance. You see only 2 of the cells are green, right? The others are not. So this is really true, and we're trying to use systems biology to work with this. I'm afraid I won't have the time at the moment, but it's quite a fancy story. It goes down to statistical mechanics and Poisson distributions and non-Poisson distributions and actual bursting. And one of the great results here is here that we look at individual messenger RNA molecules in cells. So these are different cells, and these are different individual messenger RNA molecules. So for each cell, we can count the number of messenger RNAs. We can make a distribution function of this, and then we can figure out that indeed there is this heterogeneity. You can see that here as well. Some cells have a lot, some cells have less, and we find the origin of this. And the origin seems to be in epigenetics of bursting transcription. Okay, let me cut short on this. What I wanted to tell you about is actually what systems biology is. I've been telling you that we're trying to discover principles of biology, and there's been 2 successes, one by Belyaev post factum, and then I've been telling you a little bit about progress, making my model, genome-wide metabolic map, and the fact that we can now understand noise of transcription fairly well with these methodologies. So with that, I'd like to thank you for your attention.